final presentation will be from Dr. Christine Garnett from FDA, clinical reviewer and team leader in the Division of Cardiology and Nephrology at FDA, the lead for the Cardiac Safety Interdisciplinary Review Team. She will present on pediatric extrapolation for pulmonary arterial hypertension using a physiologic biomarker of pulmonary artery pressures. Christine?
Well, thank you. So my presentation will be a little bit different from what you heard from the previous two in our session. And I will focus on clinical translational science, whereby a bridging biomarker can be used to extrapolate efficacy of a drug that's already been approved in adult population to a pediatric population with the same disease. Now, the approval of new drugs for use in children follow the same high standards as that of adults, requiring demonstration of substantial evidence of effectiveness as well as a favorable benefit-risk profile. However, there are ethical, physiological, and statistical challenges in conducting clinical trials in children, and approaches that leverage as much clinical data from this reference population to the pediatric population are important to reduce extensive testing in children. And this is why I will focus in my presentation on this pediatric extrapolation approach, as well as the bridging biomarker. And I will present a case study using PAH. Just to have some, next slide, please. So just as a standard disclaimer that my presentation is not considered official FDA guidance. And I'd also like to acknowledge contributions and insights to this presentation by Drs. Norman Stockbridge and Lynn Yao at the FDA, as well as Dr. Thomas Fleming at the University of Washington. Next slide, please. The pediatric extrapolation is an approach to provide evidence in support of effective and safe use of a drug in children when it can be assumed that the course of disease and the expected response to drug would be sufficiently similar between the pediatric and a reference population. Now, in my presentation, the reference population will be the adult population, but keep in mind that a reference population can also be another pediatric group. Next slide, please. So the factors that influence extrapolation will depend on evidence-based assumptions on the similarity between the adult and the pediatrics for the disease, as well as its response to treatment. So the first is evidence for disease similarity. This would include factors such as common pathophysiology, common disease definitions, as well as the course of disease. Evidence for response similarity would include factors such as similar pharmacology, similar response in clinical endpoints, as well as demonstrating a similar exposure-response relationship. And then there's the quality, the quantity, and the sources of existing data are also important considerations. So all these three factors will influence the choice of an extrapolation approach. Next slide, please. So the types of extrapolation approaches, you can think of them as reflecting a spectrum of dissimilarity and similarity of disease and response to treatment. So if there's a high confidence in the similarity of both the disease and the response to treatment, then pediatric extrapolation approach could include a pharmacokinetic study to show similar exposures in pediatrics to that of adults. Now, on the other end of the spectrum, if the disease is different, or if there's large gaps in knowledge, whether the response to treatment, then the pediatric approach could include one or more adequate and well-controlled trials using age-appropriate clinical endpoints or surrogate markers. Now, somewhere in between the two ends of the extrapolation spectrum is the use of a bridging biomarker, where there is high confidence in the disease similarity, but there remains gaps in knowledge on whether there is similarity in response to treatment, and perhaps there are limited age-appropriate clinical endpoints. Next slide, please. So a bridging biomarker is a response biomarker that is supported by strong mechanistic evidence and is expected to be correlated with a clinical endpoint intended to assess clinical benefit in clinical trials. And the use of a bridging biomarker in pediatric extrapolation is distinct from other roles of other biomarkers. Its role is to extrapolate the efficacy from adults to children for drugs that have already demonstrate effectiveness in the adult population with the same disease. Now, a bridging biomarker is more predictive of efficacy or the disease than pharmacodynamic biomarkers that are usually used in pediatric development programs for dose selection, as well as maybe demonstrating similarity in the disease or response. But it's not. But there's less. It's not a surrogate endpoint. There's less confidence that it directly is a measure of how a patient feels, functions, or survives. So next slide, please. Now, to justify a bridging biomarker as a replacement endpoint in pediatric trials, the biomarker should meet 5 criteria, which I have put on this slide. The first criteria is that the pediatric and the adults have similar disease. The second criteria is that in adults, the drug has substantial effect on both the clinical endpoint and the biomarker. The third criteria is that the treatment effects on the bridging biomarker capture the effects on the principal causal pathway through which the disease process meaningfully influences the clinical endpoint. The fourth criteria is that in adults and also in the pediatric populations, that the treatment doesn't have any important unintended effects on how a patient feels, functions, or survives that are not captured by the bridging biomarker. And then finally, the fifth criteria is that by proper statistical analysis, that the treatment's net effect on the clinical endpoint is consistent with what would be predicted by the level of the treatment effect on the bridging biomarker. This is very similar to the Prentice criteria, but is not as stringent. So if these 5 criteria are met, then establishing a meaningful effects on the bridging biomarker in the pediatric setting would provide sufficient evidence to extrapolate the efficacy assessment from the adult population to the pediatrics that the drug would have a meaningful effect on the key clinical endpoints. So next slide, please. So now I'll move into my case example. And this case example is how pediatric extrapolation using a bridging biomarker was used to approve the first drug for pediatric pulmonary arterial hypertension. So conducting clinical trials in pediatrics that have pulmonary arterial hypertension, I'll just call that PAH in the presentation, is very difficult. First of all, PAH is a rare disease and there's limited patients who can participate in clinical trials. It's also very difficult to conduct placebo-controlled trials because there is a prevalence of off-label use of drugs that are not approved for children, but are approved for the adults. And so it's very difficult to have patients enroll into a placebo-controlled trial. And also in pediatrics, there's a limited number of reliable clinical endpoints. A lot of the adult drugs for PAH were approved using exercise testing, such as the 6-minute walk distance. But this is really not a practical endpoint in pediatric PAH because you really can't do exercise testing in the very young children or children who have developmental disabilities. So next slide, please. So pulmonary arterial hypertension, again, is a rare disease with a diverse cardiac, pulmonary, and systemic disease that has significant morbidity and mortality. And pediatric PAH shares very similar disease with the adults, and clinical experts have agreed that the disease is similar enough for pediatric extrapolation. And so pulmonary vascular resistance is a hemodynamic measure of the pulmonary arterial pressure and cardiac output. And PVR is on the causal pathway through which the disease process impacts how a patient feels, functions, and survives. Next slide, please. So there are 10 drugs from 4 therapeutic classes approved in the US for treatment of PAH in adults. The 6-minute walk distance is a submaximal exercise test that has been used for most drug development programs to establish efficacy for new therapies in adults. And so what I'm showing you on this slide is that there is a correlation between the decrease in the PVR, which is on the x-axis, and an increase in the walk distance, which is on the y-axis. And this is a pooled analysis of 9 approved drugs from 12 placebo-controlled trials. And each individual point on this plot is an individual patient. The red is from the treatment groups, and the blue is from the placebo groups. And so what I wanted to do next slide, is just to show you, this is a pooled analysis. This next slide now shows you 2 forest plots. The plot on the left compares the relationship between PVR and 6-minute walk distance across different drug classes. And what you can see then is that across all the drug classes, there's a highly overlap relationship between the different classes, as well as overall, which is in red. And if you compare, and if you look to the plot on the right, this is looking at the same relationship between PVR and 6-minute walk distance for individual drugs. And sometimes this is an individual study, and sometimes these are pooled studies across a couple trials. And what you could see is there are also a good consistent relationship, and even with Bosentan. And I've highlighted that by the red box. So we'll go and look at more into Bosentan in the next slide, please. So in adults, treatment with Bosentan had a significant effect on both the 6-minute walk distance. It improved it by a mean of 35 meters. And it also had a significant reduction in PVR. And so when we looked at a statistical model where we were trying to evaluate the percentage of the treatment explained on the 6-minute walk distance, we found that the model showed there was about 50% of the net effect on the 6-minute walk distance could be explained by changes in the PVR. And so this intermediate result was further reinforced just by the biological importance of PVR in the disease, where we know that it's influenced by both the pulmonary arterial pressure and cardiac output. Now, in adults, as well as in long-term studies in children, there was an absence of unexpected deaths or serious adverse events with Bosentan. So we don't believe that there was any unintended effects of Bosentan in PAH populations. So overall, the adult data showed that PVR can be used as a bridging biomarker, as it met the 5 core criteria. So next to show you what Bosentan's effect on PVR in children with PAH. So we'll go to the next slide, please. So in a small uncontrolled study in pediatrics with pediatric patients with PAH who were taking Bosentan, efficacy measures were collected. Because it was a small study, there was only 19 patients, pediatric patients. They did collect some exercise capacity endpoints in there, but it really didn't show any improvements in exercise, but that's not unexpected because the study was so small. But there was improvements in the majority of the hemodynamic measures, including PVR. Now, Bosentan significantly reduced PVR, and that's shown in these box plots on this slide. So the reduction in PVR in children is shown by the orange box, and the magnitude of change or decrease in PVR was similar to that observed in adults who were taking the labeled dose of Bosentan, and that's shown by the next box, which is in light blue. And you could see that both the Bosentan treatment and pediatrics, as well as adults, were a lot lower than the placebo effects in both pediatrics and adults. And these are coming from different study in pediatrics. So this is the yellow box is a cross-study comparison with the orange box with Bosentan. But you can see that the placebo response in both the pediatrics and the adult were very similar. So we didn't expect that, the pediatrics would respond differently than the adults on placebo. So based on this study, this very small study, again, it was only 19 patients with pediatric patients with PAH. The FDA concluded that Bosentan's effect on PVR in children corresponds to a clinically meaningful effect on 6-minute walk distance. Next slide, please. So in 2017, the FDA did approve Bosentan for the treatment of PAH in children based on this extrapolation approach using PVR as the bridging biomarker. And what I've shown on this slide is the Bosentan indication in children. And which is highlighted in yellow and in orange, you can see that the labeled indication reflects the use of PVR as a bridging biomarker. So unfortunately, moving forward and trying to think about using this extrapolation approach to approve other treatments for PAH in children, we did come to a obstacle in that PVR is collected by right heart catheterization. And this procedure, just the procedure of the right heart catheterization, has been shown to cause serious adverse events as well as death in children. And so because of that, we're no longer using right heart catheterizations in pediatric trials as part of endpoints. I mean, it's still being used as part of the clinical course, but in clinical in the clinical part of it, but in terms of clinical trials, we're not using right heart catheterizations anymore. So there's ongoing efforts to be able to measure PVR or other hemodynamics, such as cardiac output by imaging techniques. And there are also efforts to develop endpoints for children collected from etigraphy. And I understand yesterday there was a question about the use of etigraphy, and I just wanted to try to address that question here, is that etigraphy would be another measurement of functional assessment, or it would be a direct measure, it would be considered a clinical endpoint. And one of the reasons I haven't, at least within DCN, we haven't seen it as a endpoint is that we still don't understand the clinical meaningfulness of any changes in measures coming out of etigraphy. And so I think there still needs to be some more work done to be able to find that, an endpoint using the etigraphy. But if we did, it would be a functional measure. So it would fall under a clinical endpoint and not a surrogate endpoint. So just, can we go to the next slide, please? So just for conclusions, I just want to point out that bridging biomarkers have an important use for pediatric extrapolation, and a bridging biomarker is very distinct from other biomarkers that are used in pediatric development programs. It's not a pharmacodynamic biomarker, nor is it a surrogate endpoint. So it's somewhere in between. And to be able to establish a biomarker as a bridging biomarker for decision-making, the biomarker should satisfy the 5 core criteria that I presented in this presentation. And I've used an example of how Bosentan was used for approving the first drug in pediatric PAH. We also have used the pediatric extrapolation with another biomarker for approving Entresto for pediatric heart failure. And we did that using the bridging biomarker NT-proBNP. So we are using this type of bridging biomarkers in pediatric extrapolation at the FDA to approve drugs for pediatrics. Next slide, please. So I guess these are my references, and I will be looking forward to our panel discussion.